If you work in biotechnology, well, most probably you would have come across E. coli in research or in industrial applications. So it begs the question, why is E. coli commonly used in biotechnology? What's so special about E. coli? Why not other organisms? Why specifically this prokaryote organism? So let's look into why. First, we have to understand the background and characteristics of E. coli. E. coli stands for Escherichia coli. It is a gram-negative bacteria, meaning if you stain it using safrarin, it will turn pink in the gram staining. And it's also facultative and aerobic. By what it means here is that it can be aerobic and it can be anaerobic, but it chooses to be anaerobic most of the time. So it has different metabolic pathways to go to. And it's also a prokaryote, it's a unicellular organism. Uh, and the reason why we like using E. coli in research is because it has rapid growth. You can grow E. coli at a rapid rate. You don't have to wait for months. You can just wait for like a few hours or days for your whole culture to grow into substantial size. And it's very easy to culture. It can survive in most media. And here I mentioned metabolic plasticity. What I mean by that is that it's very good for research because it has so many pathways. There's so many metabolic pathways you can choose from as long as you give it appropriate nutrients or the environment it needs. For example, if you limit a certain compound, for example, if you limit nitrogen, maybe it moves towards a different pathway. So it's all good for research. And last but not least, it can be genetically modified because we have the entire genome mapped out for E. coli. Moving on, I think it's important for us to look at the history and the breakthroughs as to why E. coli has been used throughout this long period of time. First, in 1885, Theodore Askerich discovered E. coli in the human colon. So he discovered this colony of bacteria or the specific type of bacteria in the colon and therefore named it after his name. In 1946, Joshua Lederberg and Edward Tatum discovered that bacteria can also exchange genetic information with one another, and it leads to the discovery of bacterial conjugation in E. coli. So they work with E. coli to figure out that bacteria can pass on genetic information through horizontal gene transfer. Moving on to 1952, which leads us to Hershey-Chase experiment, a very well-known experiment proving that DNA is the genetic material and not the long-believed protein. So here, they also use E. coli as their bacterium for experiment. Then in 1958, Matthew Matheson and Franklin Stahl performed the well-renowned Matheson-Stahl experiment to prove that DNA is indeed semi-conservative. What I mean by that is each DNA molecule consists of one old and one new strand. They performed this experiment on E. coli as well as substituting the nucleic acid component using heavy and light nitrogen, which is N15 and N14. In 1961, Francois Jacob and Jacques Monod, which is also famous for the Monod equation in enzyme technology, has discovered the lac operon model and also the gene regulation of it. So they basically prove that our operon has a operator repressor mechanism. In 1961 onwards, Marshall Nirenberg and Heinrich Mattei both deciphered or decoded the genetic code, turning AUGC letters into amino acids. So now we have the 20 amino acids. It's all thanks to the both of them. And they basically prove that mRNA can be translated into amino acids. In the 1970s, it's all thanks to E. coli that we get to develop the recombinant DNA technology, also known as rDNA technology. What I mean by that is we use the plasmid of E. coli and turn it into a cloning vector, adding the transgene into the cloning vector and forming the new recombinant DNA, which can be multiplied and be mass produced afterwards. So what I mean by that is that we cut the cloning vector, which is the E. coli plasmid, using a restriction enzyme. and then pasting the transgene, which is also known as a foreign gene, into the E. coli's plasmid and paste it together using ligase. So what we have done with that is we discovered gene cloning by inserting antibiotic-resistant genes into the E. coli plasmids, as shown in the picture in front. In 1987, when Yoshizumi Ishino and his team from Osaka University were analyzing a specific gene called IAP in E. coli, they discovered a repeating sequence in the genome of E. coli, and that led to the discovery of clustered regularly interspaced short palindromic repeats, which also laid the foundation for future research and the now well-known tool of CRISPR-Cas9. In 1997, a great milestone was achieved. The entire 4.6 million base pair genome of E. coli K12 strain was completely sequenced and uploaded onto the database. What that means is that scientists all around the world can look at this and can refer to this database to figure out the genetic functions or the metabolic pathways of E. coli. It's a very big milestone and it's useful until today. So that's it for the breakthroughs and history of E. coli. So even back in the days, E. coli was used in so many different experiments. And it's because people have been experimenting with E. coli. They figure out what E. coli could do best in certain conditions or certain environments set to ensure the production of certain compounds. Therefore, E. coli is actually used in a lot of industries. For example, the making of flavor and fragrance. One of the compounds synthesized using E. coli is limonene, which is used in perfumes, for example, because it is a volatile organic compound which imitates the smell of citrus. Aside from that, E. coli can also help synthesize diacids or diols. The di here means compounds with two of the same functional groups. For example, for diacids, the compound contains two carboxyl group. As for diols, it's basically two hydroxyl groups. So diacids, you have succinic acid, for example, which is important in pharmaceutical and also the makeup industry. And then for diols, it's also in pharmaceuticals too, especially the 1,3-propylene diol. And then there's also bioplastics, for example, polyhydroxyalkanoids, also known as PHA, which imitates the plasticity or the flexibility of plastics. But the catch here is that PHAs are biodegradable. So aside from that, we also have amino acids, lactams, and olefins synthesized by E. coli. Lactams are building blocks for specific types of nylon. Olefins are building blocks for plastics. So all this will need the help of E. coli. And as I mentioned before this, E. coli possess a very good metabolic plasticity, meaning it has different metabolic pathways depending on different conditions or different environments provided to the E. coli. And not only it's able to produce specific products, specific acids, for example, under certain conditions, we can also force E. coli to utilize other carbon sources, which you can say it's waste. Xylose waste, glycerol waste, alginate waste, all of these waste can also be uptaken by E. coli to reduce waste and to promote circular economy. You can also utilize waste proteins or C1 compounds, which also means there's only one carbon in the compound itself, for example, like methanol and formate, which helps to reduce waste and to upcycle them, turning them, valorizing them into something better. So E. coli is not only used in the industry for chemical production, but also for research. We use E. coli to investigate carbon metabolism, to figure out the respiration and fermentation of E. coli under specific conditions, for example, aerobic or anaerobic. We can also figure out how different cofactors, sigma factors, or post-translational modifications will affect E. coli's metabolism or just how it will perform under certain stress conditions. And also toxicity, how would it react to certain toxic compounds, the side reactions, metabolite damage, and also it's used as an omics tool for genomic, transcriptomics, proteomics, metabolomics, and fluxomics. Well, to say the least, E. coli is a very powerful organism. It's a model organism used in research, and it is a very potent or potential candidate for industrial chemical production too. And even though there's so many studies on E. coli, scientists nowadays are still figuring out the best ways to fully utilize its fullest potential, for example, by knocking out certain genes or by inserting certain transgenes to make it better perform under certain conditions. So there's still a lot of experiments experimenting on E. coli to extract the best potential of it. So these are the two references that I referred to for creating this video. And thank you so much for watching. Bye bye.